Hello, Sarab sir. Hello.
Hello, sir. Sharp 2 o'clock will start.
Yeah.
Good afternoon, everyone. I would like to thank you all for second session of the conference. Dear delegates, I would like to welcome our first speaker for the session, Dr. Melanie Broszat. It is my pleasure to introduce our speaker for today. Dr. Melanie Broszat is a well-known authority on HPTLC evaluation. She has been working at CAMAG as Scientific Business Development Manager since April 2014. In this position, she is responsible for scientific evaluation of different HPTLC markets, giving trainings and seminars, and building up an HPTLC expert network. Dr. Melanie Broszat did her PhD in molecular biology and environmental microbiology under the supervision of Professor Elisabeth Grauman at the Albert Ludwig University in Freiburg, Germany. Her research interest focused on the development of new analytical tools for detection and identification of microorganisms. She has worked in research as a laboratory engineer in the University of Applied Sciences, Offenburg, Germany. She has worked in quality assessment of food company in 2008 and 2009 as a laboratory engineer in the development of HPTLC methods in the food and environmental application from the University of Applied Sciences, Offenburg, Germany. She has also studied process engineering at University of Applied Sciences in Offenburg, Germany. With this brief introduction, I would like to welcome our first speaker of the session, Dr. Melanie Broszat, who will speak.
Hello, here is Dr. Melanie Broszat speaking from CAMAG.
Yes.
Welcome to our HPTLC webinar session. I hope that you can hear me in a good way if the sound is fine.
Yes, madam. Please proceed.
So my name, as mentioned, is Dr. Melanie Broszat, and I work at CAMAG since 2014 as Scientific Business Development Manager. I work in my position close together with the lab on new applications. I give trainings worldwide. And since this year, I'm also the editor of our CBS journal. Let me tell you a few things about CAMAG. So for CAMAG, the company was founded in 1958 in Switzerland. So we have now 60 years of that our company exists. And since 1958, CAMAG started with the production of aluminum oxide. This product is still sold by CAMAG. Then, since 1961, CAMAG is dedicated to thin layer chromatography. And then later on started also with equipment and automated solutions for TLC. In the 80s, the first software was developed called CATS, and currently our HPTLC software is called Vision CATS. Last year, we have launched our first 2 modules of the fully automated HPTLC Pro system, which I will later on show you during the webinar. Furthermore, CAMAG has, besides TLC and HPTLC, also a high throughput elution instrument for dried blood spot sampling. This is our management team. So with Dr. Markus Wiss, our CEO and head of sales and marketing. Then we have here Volker Waltersdorf, our CFO, Nicolas Richet, our head of research and development, and Eike Reich, the head of laboratory. CAMAG is located in the north of Switzerland and is very close to the German and French border. So also it's located next door to big pharma companies like, for example, Roche. This is our company building, the headquarters of CAMAG. CAMAG is globally active. So besides our headquarters in Switzerland, we have also subsidiaries in Germany and in US. And then we have more than 60 distributors worldwide that are serving more than 90 countries. And one of our largest distributors is Ankrom. Ankrom is our local distributor in India that has now more than 40 years of HPTLC experience. They were founded 1978. So within the next few slides, I would like to show you about TLC and HPTLC, what are the differences, and then about the concept of standardization. When we compare manual TLC and instrumental TLC, then already you can see that the conventional TLC, the manual TLC, will give some ideas about the results. It will tell you preliminary results about the zone, is it present or not. But then if you see on the other side, the instrumental TLC that is done with instruments and with a standardized methodology, the resolution of the separated zones is much better and the results will be reproducible. So instrumental TLC allows you to improve the separation and to increase the reproducibility. When we have a standardized instrumental TLC method, then transferring this to HPTLC means just a miniaturization. This can easily be done. Then we just have to reduce the volume and then take the HPTLC plates with a smaller particle size distribution. Then we need less than half of the solvent, less than a third of the time, and we can increase the number of samples. So for TLC, the standard application settings would mean 8 samples per plate, and for HPTLC, we have 15 samples per plate. The standardized TLC and HPTLC concept, or recently just HPTLC concept, was introduced also by regulations. The USP and the European Pharmacopoeia, they have released general chapters on HPTLC, how to do it. So this you can see here. For the Europeans, they have their general chapter 2A25, and the US has the chapter 203. In there, all the steps of HPTLC are well defined, that each lab can perform it to the same methodology and is able to get the same result. What CAMAG offers is a software called VisionCats that supports all these standardized methodology and also supports a work in a CGMP regulated environment and has the option 21 CFR Part 11. What was defined in the general chapters? So the first were the USP, the US Pharmacopoeia. And in there, they have defined the plate layout, that we use 20 by 10 plates, that we apply 8mm bands, and that we develop up to 70mm. Then also it's defined a minimum distance to the plate side, that we have here 20mm. It's defined to apply bands by spray-on technique. And if you compare here spot application and band application, then you can clearly see that spray-on technique leads to better resolution of the separated zones. And also you have the opportunity to apply any solvent by adjusting the dosage speed to your sample. Then it was defined the band length. So in the past, it was written between 5 and 10mm. This is not really well defined. Then somehow the middle was selected, and now it's defined to 8mm. Also, the distance to solvent level was defined, that we have a distance from the solvent level to the application position of 3mm. Developing distance from application to front was defined to 6 centimeter. And the chamber configuration, if nothing else is mentioned in an individual monograph, then the gold standard is to develop in a saturated twin trough chamber, saturated with filter paper, and then do the development. The relative humidity here, also the standard would be to first activate the plate at a relative humidity of 33%. This can be achieved by saturated magnesium chloride solution. Here a summary. So the plate is defined, 20 by 10 HPTLC plates with a fluorescence indicator, application of 15 tracks with 8 millimeter bands, 8 millimeter from the lower edge and the first track at 20 millimeter, then conditioning with saturated magnesium chloride to adjust to 33% relative humidity, and development from the bottom of the plate, 70 millimeter. That means from application to front at 60 millimeter of developing distance. And saturation is done 20 minutes with filter paper. Solvent level is kept to 5 millimeter, that we have a distance from solvent level to the application position of 3 millimeter. Why we need standardization? Standardization leads to reproducibility. If we compare here the results obtained from here Ganoderma from 2012 and then compare it to 2016, then yes, we are reproducible even after several years. And we are also then able to compare results obtained from different blades, even from blades that were analyzed by different labs. And those data can be compared in a so-called comparison viewer. So this is now one plate with semicentrifuga, another plate, third plate, and fourth plate. And then the software allows us to do a rearrangement of selected tracks. So if we extract these tracks from the different analysis files, then we can put them side by side on a virtual plate in the comparison viewer and cluster them, rearrange them to similarities and differences. CAMAG is the world leader in planar chromatography, and we offer devices and instruments for TLC, HPTLC, and now also for HPTLC Pro. When you look on the website, then the TLC segment is more for the manual devices and used in R&D, QC for education, usually for qualitative tests. Then in the HPTLC segment, you will find everything to perform qualitative and quantitative reproducible analysis in R&D, QC, education, or smaller size companies. So this is where most customers will find themselves. And then the HPTLC Pro, this is the first fully automated sample analysis and evaluation system for routine quality control done by HPTLC. And this is addressed to high sample throughput in QC. So any quality control lab that has a high sample throughput will be happy to have an automated system. The VisionCAT software that CAMAG offers is easy to use. It has an intuitive handling and it guides you through the analysis. So when you look here on the screenshot how the software interface looks like, it's a graphical interface. And we have for each step different options for the application step, for development, derivatization, and detection. So here on top, you can see the modules of the HPTLC Pro. Then you can see HPTLC instruments, and you can also see manual devices. The software allows you also to document manually performed steps. So for reporting, all steps are documented and then everything is traceable. That also means that at any time point, you can upgrade your existing TLC and HPTLC devices or instruments to the next level and give it and put inside the automation. By clicking on the icons in the sequence you want to perform the inside of the analysis, you will create here a method. So this would consist of a clean plate image application with the module HPTLC Pro module application, then automated development in HPTLC Pro module development, detection with the visualizer, and detection with the TLC scanner. Just by clicking on the icons in the sequence you want to perform inside of your analysis. HPTLC can be used for quantification, yeah, for sure. And also, we have nice features in the software allowing you to do profile comparisons. So from the same plate, also from different plates. The software features allow you to do single or multi-level calibrations. And then also you have the choice of other evaluation features like limit tests or related substances. Any VisionCats licensed user has then also access to our method library. And the method library has methods that are ready to use. Each method entry will have an instrumental method. It will have a method document, so an SOP that describes how to perform the test, and an image comparison file with reference images that our lab has created. So the lab has done for each method analysis of reference substances and reference materials, and also then of related species. And in this collection, you can put your own data and then compare if you are able to get the same result, the same RF value. And this gives you also a nice feature to compare side by side your samples with the samples that we offer you for free by these digital reference images. HPTLC has many benefits besides other analytical techniques. One of the benefits is the visual evaluation. So we can compare visually many samples on the same plate, but then also with the comparison, the samples that are originating from different plates. HPTLC is reproducible and offers also to reliably quantify. Also, HPTLC allows you to analyze from one separation multiple times. So once you have done the separation, all components are stored on the plate and are accessible for multiple detections after development and after derivatization and after effect-directed analysis. So this shows you now a comparison file of different curcuma longa and other species side by side. And you can see them after development under UV254, UV366, after derivatization with anisaldehyde under white light and under UV366. So of each sample, one separation, and then 4 different detection modes. And as mentioned before, we can run many samples in parallel. So we are not a sequential technique. We are analyzing many samples side by side on the same plate. Another benefit of HPTLC is the easy and rapid substance confirmation by MS coupling. So selected zones can be directly eluted from the TLC and HPTLC plate and transferred to a mass detector for substance confirmation. This is now a typical setup of an HPTLC process. So for application, our ATS4, the automated sample applicator, and for development, the ADC2 that has a twin trough chamber. Development can be done in a saturated or unsaturated tank with or without filter paper. The humidity can be adjusted with molecular sieve or saturated salt solutions, and the development is detected during the chromatography. It's monitored, and then when the migration distance has reached the set migration distance in the software, then the chromatography will be stopped, the plate moves up and is dried in an airstream. So here, this is fully software controlled. Then for derivatization, we have an automated spraying device that uses for each HPTLC plate just 3 ml of reagent. And the reagent is distributed as very fine mist. So we have micro droplets that are produced and then settling homogeneously on the plate. This gives you comparable results to dipping and has the advantage that it does not consume 200 ml like for immersion, and that each time you will use fresh reagent, just 3 ml per plate. For detection, we have for quantification and for UV spectra, the TLC scanner. And for mass confirmation, for MS spectra, we have our TLC MS interface. This would be the results of the detection. And here, the results of a typical mass spectrum obtained from a plate. The TLC MS coupling works like this. We have our elution head. The position of the selected zone is here selected. And then with any LC pump, you can directly elute the zones from the plate and transfer it to any mass spectrometer. Here is a small exchangeable filter that the solvent that elutes the zone and is transported to the source will not also transport the silica gel particles. So only the dissolved substance zone will be transferred to the mass spectrometer. And here you can see our TLC MS interface, the standalone device that is not software controlled in a typical LC-MS lab environment. The next level is our HPTLC Pro. And this is the next level of HPTLC. So here with the HPTLC Pro, the plate will be transported from one step to the next one via a conveyor. And then no manual intervention during an HPTLC process is required. We have also here application, development, derivatization, detection, and coupling to mass spectrometry. HPTLC Pro offers fully automation, leads to highest reproducibility, and supports high sample throughput. So the system was created with the opportunity to autonomously apply up to 75 samples on up to 5 plates without any manual intervention. The plate is located in a carrier, and this carrier is recognized by the system. So at any time point, the location of the plate is known during the process. And via the conveyor, the plate is transported from one module to the next module. Here a short teaser video. So we have the application step with a laser controlled needle distance. We have the development in a chamber with a optimized chamber geometry. Derivatization is then software controlled. And we have hyperspectral imaging for the detection and our VisionCAD software for the data treatment. MS coupling is then also software controlled. So selected zones will be automatically eluted with the HPTLC Pro module MS interface. When we look for the already available modules, the application, then what's new? Now we have a laser controlled needle distance. So the distance of the needle to the surface of the plate can be optimized, and we have done this for the most common solvent systems, most common solvents. And then prior to application, the distance is controlled. So it is kept at the same distance. Then we just apply the needed volume. So the syringe will fill up to the required volume, and it's always quantitative. We have two rinsing solutions, and the second rinsing solution is then also a little left in the top of the syringe to create a separation bubble to prevent cross-contamination. The autonomous application of up to 75 samples is supported by the sample rack that can have 75 vials. For HPTLC Pro, HPTLC glass plates have to be used, and just in the format 20 by 10. The syringe is providing, in the syringe, you have a volume of your sample between 1 and 8 microliters. So we have a 10 microliter syringe that comes with a serial number. All the modules for the HPTLC Pro have the same design. So they look from the outside identical. And we have also from left side the option that we can manually load a plate if we don't have the plate storage for automatic feeding. On top, for the application module, there are 3 bottles, 1 waste bottle, 1 rinsing bottle, and a second rinsing bottle. The plate is placed into a carrier, and we have a sample rack with 75 vial positions. For the module development, so what is new here? We have now for the module development a low chamber volume. It's not anymore a twin trough chamber. And this allows us to better control the gas phase. The distance between layer of the stationary phase and the chamber wall is reduced to a minimum. And this speeds up also the conditioning. So we have a fast activation and preconditioning of the stationary phase. And what we can also do is that we circulate the gas phase during chromatography. So this is leading to really new options that have never been available before. The gas phase during development can consist of the same mobile phase system, the same developing solvent, but it can also consist of a totally different solvent or solvent mixture to further optimize the separation. Then also the volume of the developing solvent during chromatography is controlled. We will always have the same solvent level. It's refilled to keep it on the same level. And the developing solvent between different methods, between different plates, is changed without human intervention. We have 3 solvent bottles for a maximum of 3 different developing solvents. The gas phase is kept under control and can be used to further optimize the separation. So also here from the outside, it looks identical. And we have on top 5 bottles. So 1 waste bottle, 1 bottle for activation. You can fill in there the molecular sieve for trying for full activation of the plate or different saturated salt solutions. Then we have 3 solvent bottles. Sorry. And also here, you can also have this not connected to other modules and then do the manual loading from the left side for this instrument, for this module. So what about people having developed their methods in twin trough chamber in the ADC2? they can transfer the methods also to the HPTLC Pro module development, and then it's not required to use a filter paper. And by this faster preconditioning and activation, you will also speed up the whole analysis. So the chromatography will be faster compared to twin draft chamber, where you have a minimum of 20 minutes of chamber saturation, and also you have the activation step. This on the left side shows you an example about the separation of ginkgo extracts on normal phase in the ADC2 and in the HPTLC Pro module development. On the right side, you can see an example on reverse phase. So here, fixed oil separation in the ADC2 and in the HPTLC Pro module development. We have in our instruction manual then a guideline that shows you how to transfer the methods. So in principle, for the normal phase, preconditioning will lead to a decrease of RF values and conditioning will increase the RF values. And on the reverse phase, it's the other way around. Preconditioning will increase the RF values and conditioning will decrease them. So during this year, we will launch the module plate storage, and this is then required to automatically feed the HPTLC Pro system with clean plates. Up to 5 clean plates are in one compartment in the upper part of the module, and then up to 5 process plates are stored in a separated second compartment in the bottom part of the module. Also, for sure, this module is software controlled by Vision Cats. So CAMAG has its general software for all steps of TLC and HPTLC Vision Cats for now and for the future. So for whom was HPTLC Pro designed? For all routine QC laboratories with a large number of samples per day. Applications can be for efficient limit tests performed in the pharmaceutical industry, for food testing, any contract laboratory that provides analytical services, and all companies that need a fast screening method for analysis of multiple samples, especially those with diverse and complex matrices. So that was about the instrumentation, and now we will talk about HPTLC comprehensive HPTLC fingerprinting and also about HPTLC applications. The first I would like to show you is comprehensive HPTLC fingerprinting, that you can use one method to check for the identity, purity, and content. So now we have our raw material or our product, which is extracted. Then we have our sample solution. Then we apply the sample, we do the HPTLC process, and then we generate our data. And there we have now one separation, and then before derivatization, 1, 2, 3 detections with the visualizer. So by image documentation, under white light, UV254, UV366. Then after first derivatization, under white light and UV366. And in some cases, we can do even a sequential derivatization. So here first with natural product reagent, and then a second derivatization with anisaldehyde, and then do again image capturing under white light and UV366. So one separation and 1, 2, 3, 4, 5, 6, 7 results just by the image capturing. And this set of data can be used for evaluation, for a real identification. But besides identity, the HPTLC fingerprint also contains information about the quantity. So besides images, we can use image profiles, and we can use densitograms recorded by the TLC scanner. And all of this can be used then to check identity, purity, and content. For HPTLC Pro, we will then have soon the hyperspectral imaging. So here a short animation. One sample separated and then detected multiple times. Hyperspectral imaging means that instead of just RGB or single wavelength, we get in each measured point on the plate the entire spectrum. So for each point on the plate, we get the full spectrum from 200 to 1000 nanometers. And this multiple times if we do a hyperspectral detection after development and after derivatization, and maybe also after effect-directed detection. So HPTLC Pro and HPTLC also now allows you to define acceptance criteria obtained in different plate stages. One sample separated and then detection prior and after derivatization with different detection modes. That means that we can create for each sample acceptance criteria and create like a template for automatically evaluation. If the sample matches with this template, then this would mean the sample has passed. If it doesn't match with the template, then it is a fail. Comprehensive HPTLC fingerprinting. Our lab has worked a lot on this topic, and especially here, Deborah Frommenwheeler, who is doing her PhD on this topic. So she has now developed methods that can do with the fingerprint methodology, also give you the information about purity and content. One example is here the case study of Angelica, and the ID method can clearly identify 28 Angelica and related species. They can be distinguished because all of them have a different HPTLC fingerprint. But also this method allows you to detect mixtures between different species. Here in this, you see 1% of Angelica acutiloba in Angelica gigas. And here you see 1% of Angelica gigas in Angelica acutiloba. Then also with two other species, Angelica sinensis in Angelica gigas and Angelica gigas in Angelica sinensis. So 1% of a mixed species in the other one can be seen in the fingerprint. And the same method can be used to quantify the minimum content. So one method gives you all the information about purity, identity, and content. This shows another example about comprehensive fingerprinting, and here for ginkgo products. So the lab has done a market study and tested many different ginkgo samples. So in total, they have tested 59 products and 48 of them were of poor quality. So they either showed one or more adulterants. If you look here on the reference extract and then here on the different products, then the first two, they look like they should. They are similar to the reference material. If you look on all the others, then you can see for the tracks 3 to number 8, they have a very high fluorescent yellow zone on the upper part of the chromatogram, which was caused by addition of quercetin. And some here on tracks 9 to 11, they show a very intense routine band, but then a very faint or no ginkgo fingerprint in the back. So this can be caused by adulteration, that a cheap flavonoid is added because of economically driven reasons. And the total flavonoid content measured by photometry will pass in this case. It will just tell you, okay, fluorescence intensity is fine. But then HPTLC would have told you, no, it is not fine because it's just added a cheap flavonoid. And there is, in some cases, no ginkgo at all in the product. The method to do this was published, and also you have an application note on the CAMAG website. So here is the result about the analysis of these 59 products, not only about the identity, also about the purity, and about limit tests for the added flavonoids or for the content of flavonoids. So in this upper part, you have the identification of the ginkgo products. Is it ginkgo or not? Then you have here the purity. Is there the addition of different species of different herbal materials? So the first one shows you here the test for Sophora flower. Then this one is the test for Sophora fruit. And this one is the test for buckwheat herb. With the HPTLC fingerprint, you can have in the one method check for 3 different common adulterants. So for 3 different plant species that are sometimes added to, because of economically driven reasons. And here in the limit test, on the UB254, you can check the amount of rutin. If it's above, then you know, okay, there was the flavonoid added. And on the UB366, after derivatization with natural product reagent, you can check for the content of the quercetin. And this shows now the scheme, how to proceed. So prior to derivatization, you have the check of buckwheat herb. Prior to derivatization, you have here the check of Sophora fruit. And also Sophora fruit, you can have a second confirmation after derivatization on the UB366. And then after second derivatization on the white light, you can see if there was Sophora flower added. One separation, different detections, and 2 derivatizations, that gives you then all the information about your sample composition. So now we will have a look at the different application fields for HPTLC. This is a screenshot of how it is segmented on our website. We have the 8 application fields, herbal drug, etc. And for each of them, we have application notes and case studies on the website. So you will find many methods and examples on the CAMAG website. Please have a look at them. Now for a few, I can have the time to present them. So let's get started with the cannabis testing. With HPTLC, you can have just by an application development and detection step, the answer for a limit test is the THC limit above or below the allowed amount. So an easy limit test can be performed that tells you quickly above or below the permitted level. Or you can do a multi-level calibration and then also say exactly how much of the cannabinoid is inside of your cannabis sample. For confirmation, prior to derivatization, you can also do UV spectrum, record the UV spectrum, and then overlay the UV spectrum from your sample with the reference spectrum and see if it has a match. So just by these 3 steps, application, development, and detection, you have these 3 options or these 3 possibilities. Then if you add 2 more steps, 1 step for derivatization and another step for detection, then you can also get the information about the identity. Same plate, but then sprayed with the with vanillin reagent and then detected under white light you will get the information about colored zones plus RF value, which cannabinoid is present in your sample. And the intensity will tell you also on how much you have inside. This shows you here on reverse phase the separation of 14 different cannabinoids. And if you like, you can add another step, the MS coupling with the TLC-MS interface, or soon also the HPTLC Pro module MS interface, and then elute selected zones and analyze them with a mass spectrometer. Can be single MS, can be tandem MS, QTOF. So TLC and HPTLC can be coupled to any MS system if it has a AC or APCI source. So soon we will have then also here the module MS interface, and we will have the hyperspectral imaging, and then the whole cannabis testing methodology can be done automatically, even overnight, that the person prepares on one day several plates, several samples, put them to the HPTLC Pro system, feed the plate storage with the 5 plates, and then the plates are automatically processed by the system. The analyst comes back and evaluates the data. This you have seen before. Now, other examples, HPTLC allows you also to do an efficient screening of dietary supplements for synthetic drugs. This is a method that detects 9 synthetic drugs with one method. And we have here a system suitability test with 3 of these 9 substances to check the reproducibility, the chromatography, if the conditions have been reproducible. So we will check for each substance of the system suitability test the RF value with our defined acceptance criteria. And then here you can see this 9 separated substances. This method is efficient because you can again analyze many samples in parallel and not sequentially. And market studies have been analyzed by our lab, and some have shown adulteration. Here, one sample has shown adulteration with phenolphthalein and also sibutramine, which was not declared on the product. HPTLC also allows you to screen forensic drugs from 6 substance classes with 1 mobile phase system. So this was a bachelor project in 2018. And with this method, you can analyze the most common street drugs, plus the cutting agents. We have an SST, and with 1 separation, you get 4 analytical endpoints: the RF position, UV spectrum, mass spectrum, and color after derivatization. This is also available on the CAMAG website as application note. I cannot go into detail for each application, and we have many, many more on the website. So please also have a look at them. Then HPTLC is also a good tool to check inks. The inks can be tested for document forgery, who has done the signature. So the Secret Service in US, they have analyzed more than 8,300 coded samples together with CAMAG. And this is now here a nice tool for a fast fingerprint analysis. Has it the same pattern or not? Another example from the USP is about the fixed oils, and this method is also in the European Pharmacopoeia, where it's called fatty oils. So with this method on reversed phase, you can identify, is it an olive oil, a corn oil, or another vegetable oil. This was implemented in 2015. This method shows you the adulteration of quite expensive essential oils with cheap vegetable oils. So this is now not on reversed phase. This is now separated on normal phase and then derivatized with anisaldehyde. And then if you have analyzed many samples, you will fastly see if there is an additional zone here on top. Whenever there is a zone at this position in the fingerprint, then you know this was mixed with a cheap vegetable oil. And then you go back to this method that I have shown on the previous slide and put the sample on reverse phase and can see which vegetable oil was added. Here on back, you see the GC method for the essential oils, and this would not have shown this adulteration. For this, you would have needed to do another second analysis, another methodology. So the HPTLC shows you the identity and if there is an adulteration, and if you see here a zone that was caused by an adulteration, then you can confirm this by putting the same sample on the reversed phase. Then this example shows you another example, shows you how to do identity of different caffeine-containing botanicals and how to determine the caffeine level. So with one separation, you get again the discrimination of caffeine-containing botanicals and also the amount of caffeine, or you can also use it for a limit test for decaffeinated products. Under UV254, you detect the caffeine, and also with a scan at 273, you can precisely quantify. Then after derivatization with anisaldehyde under white light, anisaldehyde plus natural product reagent, you can discriminate cola and guarana. And after this double derivatization with natural product reagent and anisaldehyde under UV366, you can discriminate tea, mati, and coffee. So one method, two derivatization, several detections, and you get all this information out of one separation for each sample. This is also available as application note. Also, HPTLC is a useful technique for efficient cleaning validation protocols. And this example is from Sanofi in France. So they have also written a CBS article about their application. They just put the rinsing solutions after cleaning the vessels on the HPTLC plate without sample preparation, develop the plate, and detect under with the scanning densitometry in the UV range. And then immediately they have the result within less than an hour. Is the cleaning procedure efficient or not? So is after the third rinsing, the API below the specified amount. Any colored sample is also predestinated for HPTLC. So this method shows you the screening for 15 illegal dyes. They can be added to pepper, paprika, to different spices. So this is a nice example of screening for illegal dyes in, for example, spices or other food matrices. And HPTLC can be used for sugars and sweeteners, natural sweeteners like stevioglycosides. All sugars and sweeteners show just small or no UV absorbance. So with derivatization, you can make them detectable and then quantify them sensitively. And HPTLC can deal with these polar substances easily. Then food contaminants, like for example, the aflatoxins, they show a native fluorescence. So all compounds that show fluorescence, either native fluorescence or fluorescence after derivatization, can be sensitively detected by scanning densitometry and later on also by hyperspectral imaging. This shows the separation of the 4 flavonoids. So with this, you can detect with this separated separated zones, you can quantify those 4 aflatoxins down to the low ppb range. What also HPTLC offers you is an analysis of really complex metrics like heavy petroleum products, bitumen. HPTLC plates are used only once. Therefore, if there is a high matrix load and your analytes get separated out of these heavy metrics, then this is not an issue for the next sample. On a column, you would have a problem if there is something remaining from the separation of your previous sample. If you have a carryover, then you will have issues with some ghost peaks or with some separation that is not really looking good. HPTLC, each sample is applied on a new position on a plate. Therefore, matrix is not an issue. And here, heavy petroleum products can be fractionated to the fractions about asphaltanes, resins, aromatics, and saturates after here addition of a fluorescent reagent. And then you can say, get the sum parameter, SARA analysis, how much of each of these groups is inside of my heavy petroleum product. In general, lipids can be nicely separated on silica gel. So this shows you an example about the separation of polar and apolar lipids on one plate by the AMD technique. The automated multiple development allows you to develop a plate not only once, to develop with different composition of developing solvents to different migration distances. And by this, you can increase the number of baseline separated compounds up to 40. So finally, I would also like to show you the option that you can do effect-directed analysis after HPTLC separation. The chromatography is the same. So you apply your sample, you develop the plate, and then after this, you can apply enzyme solutions, substrate solution, or also you can apply bacteria or yeast cells. Then usually you have an incubation step, and after this, you can do the documentation and evaluation. The effect-directed analysis on the plate can be done because silica gel is quite inert. After development, the solvent is gone. So the toxic solvents, they are not disturbing the growth of bacteria or enzymatic reactions. And also the silica gel is quite neutral, or you can even adjust the pH if the organism likes to have it a bit alkaline. You can dip the plate into a buffer solution. So then after the enzymatic reaction or the growing of bacteria or yeast cells was done on the plate, you have the readout and you get then the information about the effect and besides the chemical properties that you have obtained already before. So this is a very strong tool. And these are common assays. So I'm running out of time, therefore maybe you can later on check out which assays are available. And this would be a typical approach. So we have application, development, detection without any enzymatic or bacterial assay, then application of the enzymatic assay or the bacterial or yeast assay and doing the readout. This setup is very useful in research. And the research is, for example, of new active pharmaceutical ingredients. So you immediately get the information about a separated substance zone and its effect. Further information you can find on the CAMAG website. So we have here on the CAMAG website in the section Education, our customer magazine CBS. This is released twice a year and gives you application examples from customers and also from our lab. Then we have on the website our database CCBS. It contains more than 11,000 abstracts just dealing on TLC and HPTLC applications. This database can be used for free, so use it. For each entry, you can search for keywords, and for each entry, you will get the information about which plate to use, which mobile phase system, is derivatization needed, and which detection mode. So whenever you have an analytical problem, maybe the CCBS database will give you a good idea about how you can solve it. Then these are links to sources of methods. And what I would highly recommend is the HPTLC Association. There you can also apply to be a member. It's a non-profit organization that wanna harmonize HPTLC worldwide. And there are methods on identification, and other methods will also be implemented, as well as an HPTLC atlas with reference images. Then we have a YouTube and LabTube channel where you can find videos on applications and product videos, and application notes as well. So also, if you have questions, feel free to write an email. And therefore, now I would like to thank you for your attention and hope that you did enjoy the HPTLC session.
Thank you, Dr. Melanie, for the wonderful session. What makes an analytical technique dynamic is its higher level of automation and diverse application. The insight you have provided will help us to explore new areas and dimension in HPTLC evaluation. Now, since we are running out of time, I think all our delegates are very inquisitive, and the team Encrom has settled most of the questions. Still, if there are any questions, delegates, just write back to us, and Team Encrom will definitely get back to you. So, on behalf of everyone connected here, I would like to thank you, Dr. Melanie, again for the informative session. I would also like to thank Dr. Charegaonkar, Dr. Akshay Charegaonkar, and Team Encrom for their continuous support. Thank you, Dr. Melanie. Okay, now let's move on to the next speaker. I'm delighted to introduce Dr. Stephanie Harden from Waters Corporation, Switzerland, who will tell about enhanced approaches to analytical method development and management in support of forthcoming ICH guidelines. Dr. Stephanie Harden did her PhD in chemistry from University of Bristol, UK. Following her postdoctoral research, Stephanie moved into industry and joined Waters. She is currently in Waters for 20 years and is responsible for growing Waters' European small molecule pharmaceutical business. In her present role, Stephanie worked with regulatory bodies and industry thought leaders to promote modern approaches in developing and controlling pharmaceutical products. Dear delegates, I invite Dr. Stephanie to begin with our session.
Good morning, thank you. Can I just confirm that you can hear me?
Yes, ma'am.
Great, thank you very much. So yes, I'm very happy to have been invited by the organisers to present today at the conference. And both myself and my colleague, Guillaume Bechade, who will present after my presentation, we're very happy to present on behalf of Waters Corporation. And in case you're not aware of Waters as a company, I just wanted to give you a couple of slides of brief introduction to us. So Waters Corporation is a global company focused on specialty measurement, and we provide a portfolio of systems to analytical technologies to help customers to improve the way they're working. We're renowned for our innovation, our applications expertise, and particularly for our focus on customer success. And our portfolio of analytical technologies can be applied across a number of market areas, including pharma, materials science, food and environmental, clinical, and biomedical research. So yeah, we very much focus on our customers, on our customer success, and helping customers be better at what they do and improve the results that they get in the endeavors that they're working in. And in this presentation, I'm very happy to give you some information on new approaches to analytical methods, new approaches which are advocated in the proposed guidelines from the ICH and the USP, which are currently in review at the moment. And in the presentation today, what I'd like to do is to give you an overview of some of the challenges which we currently have when we're using traditional approaches to developing and managing analytical methods. I'm particularly talking today to methods related to the pharmaceutical industry and guidelines which are related to the pharmaceutical industry. We're going to look at some modern concepts around life cycle management of analytical procedures. And particularly, I'd like to focus on quality by design concepts and how they're applied to analytical methods. We'll look at some of the benefits of hyphenated liquid chromatography and mass detection. And I have a small example of Bastine API, which is a method in the pharmacopoeia, which was redeveloped. And I'll show you the benefits of applying a quality by design approach to developing methods. Following that, we'll look at the importance of control strategies and also the importance of continuous performance verification of methods over the whole life cycle of the use of an analytical method. And I'll finish with some sources of further information for you in case you're interested in learning more about this topic. So to start, the way we typically have worked in the past, the traditional approach to managing analytical methods has been fairly disjointed. So we start by developing a method, and typically we develop a method based on our experience or using an ad hoc approach, taking a column that we know well, a C18 column out of the drawer, and doing a fairly unsystematic approach to method development. And this would typically result in methods which were fine, but not as robust as they could be, and perhaps not as fit for purpose as they should be. Methods are then validated based on, as a kind of checkbox exercise, and transferred to a receiving lab. And then finally put into routine use, of course. And that's typically where things stop. They go into routine use and the method isn't really touched for, in many cases, for many years. So this is the traditional stepwise approach to method management, method development and method management. And there's very little connectivity between these different stages, very little in terms of feedback or improvement of methods over time. And the downside of this is that it leads to a very limited understanding of the method, a method which is very much more difficult to troubleshoot and to transfer as well. We have very limited understanding or control over the sources of variability around the method. And so we're very much more likely to see failures once that method goes into routine use. The lack of communication across organizations is also a challenge today. And as we see a greater globalization of companies, a lot more outsourcing of different parts of the drug development cycle to contract developers and manufacturers, there's a lot more of a disjointed situation. And generally, this lack of communication across organizations leads to, again, limited understanding of the method and much more difficulty in troubleshooting and controlling that method. And finally, continuous improvement. There's very little encouragement at the present time for continuous improvement of existing methods. And one of the reasons for that, of course, is the lack of regulatory guidelines around quality for analytical methods. So the current framework which we have in place doesn't encourage users to understand their methods. It doesn't encourage appropriate control of methods and or the reduction of sources of potential variability. So this is very key, because how can you ensure the quality of your analytical method if you really don't understand it and you can't manage the potential variability around that method? And then, of course, the upshot of that is this potential cost of poor quality, which can then reflect on the industry. And there are huge costs to industry in terms of fines, losses, delay to market, wasted time in reworking methods and trying to understand where problems came from, and so on. And a little while ago, one of a well-known quote from Michael Kopka from the FDA was that 65% of all drug shortages are caused by manufacturing and quality issues. Of course, contributing to that are quality issues caused by poor methods, of course. So there are many reasons why we should be improving our approach to analytical method development and management. Now, there are some forthcoming changes to the regulatory guidelines around analytical methods. And one of those is the new USP Chapter 1220, which is in review at the present time. This, as you can see in the slide, is a stimuli paper, which was first brought out in February 2017. And this outlines the new approach, the modern approach to developing an enhanced methods, enhanced management of methods. As you can see here, just to give you a brief description in the abstract, the guidelines say that an analytical procedure must be demonstrated to be fit for its intended purpose. It's useful to consider the entire life cycle of an analytical procedure, i.e. its design, development, qualification, and continued verification. The current concepts of validation, verification, and transfer of procedures address portions of the life cycle, but do not consider it holistically. The purpose of this chapter is to more fully address the entire procedure life cycle and define concepts that might be useful. And the approach is consistent with the concept of quality by design. And this is very important because quality by design and building quality into what we're doing was introduced quite some time ago now, of course, for processes and procedure and manufacture of pharmaceuticals. But up to this stage, there are no guidelines which talk to the application of quality by design concepts to analytical methods. So these new guidelines which will come out are really doing that. They're saying that they're advocating the application of these quality by design principles to the development and management of analytical methods. And in this presentation, we're going to take a little bit of a look in more detail at the analytical procedure life cycle. So these USP-1220 guidelines are expected to be in the Pharmaceutical Forum as of September the 1st, 2020. So this is coming up now fairly quickly. And one diagram which really shows very well, I think, the proposals within USP-1220 is this one. So these are the method life cycle stages. And instead of that traditional approach that you saw right at the beginning of the presentation, where the stages of the method are very disjointed with little connectivity between them, the new proposal is a very much more streamlined approach with like a cyclic circular approach, which involves a lot of feedback and a lot of continuity between the different stages of the method's life cycle. The guidelines propose that we start with this analytical target profile. So we start by defining the analytical goals, the goals for developing our method. And the goals, of course, for our method need to be based on the process. Which aspect of our process for manufacturing a drug need to be controlled to ensure that we are producing a quality product. So we start by identifying the goals of the method. In stage one, we design and develop the method. And this is a fairly complex process of gathering knowledge, background knowledge about our sample and the compounds, conducting a full risk assessment of the method or related to the method, and putting control strategies in place around that method. And we're going to look at some of those aspects in this presentation. Following the design and development of the method, we move to stage two, which is the method performance qualification or verification of the method. And the verification needs to be based on our initial goals or the analytical target profile. So what we are verifying is very much related to the goals of the method. And then in stage three, we are putting this method into a routine use, and we start with a continuing procedure performance verification, which means that when the method is in routine use, it doesn't just stop there. We continue to monitor the method using trending, for example, over time to ensure that that method is still working, and that it's, of course, to ensure that that method is still fit for its intended purpose. If any changes are required or if improvement is needed, then that has to take place. And then, of course, we'd go back perhaps to stage two or to stage one to implement those changes. Second guideline, which I like to mention, is from the ICH. So this is ICH Q14. And this is another guideline which will be harmonized with USP-1220, which talks to the analytical procedure and the life cycle of the analytical procedure. So this is accompanied by a revision of Q2R1, which will then be Q2R2. This is the guideline referring to analytical validation. And all of these changes mean that there will be a harmonized recommendation to how we, moving forward, design, develop, and continuously monitor analytical methods. Again, the ICH Q14, as you can see on the bottom here, highlights the fact that it advocates the use of quality by design principles in the management and design of analytical methods. And this guideline is expected to be published in 2021. And there's a lot of information there in those guidelines, but one of our collaborators, Phil Borman from GSK, did a very good job of just in basic terms describing the changes to be expected as a result of these new guidelines. And he said, this is really trying to develop a framework, good ways of working, and tools for analytical scientists really to show what they know about the analytical procedures they develop, to capture the information, to transfer that information to the long-term users of analytical methods, and really build that knowledge as we go. So we have good control across the life cycle of that analytical method. So right till that analytical method goes out of use, till it's not used anymore, it's retired, it has to be managed and ensured that it's still fit for purpose. So what's different? The life cycle management concepts promote the identification of parameters that contribute significantly to method variability. So it's very important to do risk assessments to understand those factors which will affect variability. We need to put control strategies in place to minimize risks where they're identified. And eventually, this will lead then to robust methods that are less susceptible to failures and provide potentially some flexibility to regulatory changes. So we heard about the application of quality by design principles to the management of analytical methods across their entire life cycle. But we can also apply quality by design principles to developing the analytical method itself. So essentially that means that you're designing quality into your method. And some of the goals of applying this approach to methods are shown here. So when we start developing a method, we're not basing our information generally on our predefined information, on information we currently have about other methods we've developed. The idea is that we go in with a blank sheet of paper and we look at this with a fresh perspective, and we're not assessing the situation based on previous results. We perform a design of experiments, and this means that we are evaluating the most influential parameters, but also importantly, identifying the interaction between different parameters. And this allows us then to optimize the best conditions for our method. So it's a risk-based approach. And in the end, we should obtain knowledge of how that method performs and understand the limitations of that method. So understand the edges of failure of that method. And if we apply a quality by design approach to developing our analytical method, and particularly if we use specific software, there are a number of softwares available on the market which are designed to help us develop methods using quality by design concepts. And the reason for using software tools for developing methods using quality by design concepts is that it simplifies the process quite significantly. If you imagine you're comparing a number of different parameters when you're developing a method and you want to identify the interaction between parameters, this is much more easy to do using statistical software and modeling software. Eventually, when we do this, we can define a region of chromatographic space based on our experiments within which we know that our analytical method will be robust, and within which any changes within that design space won't affect the quality of the analytical result. So this is an example of a graphical representation of a design space. And once you've carried out your analytical method design and development, and you've tested a range of analytical conditions, and in this example here, we're looking at final percentage organic versus gradient time, you'll have a good understanding of the method, and you will have tested. You can see here the knowledge space, that's the area you've tested. And in the brown circle, this is your design space. And this is the area within which you know that your method is going to be within your acceptable conditions. And then within that, you would select an area to validate, or a method to validate. the NOR is representing the normal operating range of the method in practice. And the eventual anticipation is that once you've carried out an approach like this, a quality by design approach, and you have assessed the robustness of your method using under a range of different conditions, and you know that your method will be robust within this particular space, that there will be some regulatory flexibility to move around within this design space. Now, we mentioned the importance of assessing variability of methods, really understanding variability and where potentially variability can arise related to your analytical method. So in this slide, you can see that there are many areas of potential variability. That may be related to sample preparations, your methods, materials, and instrumentation. And in fact, many aspects of potential variability, or many areas where methods fail typically, is related to sample preparation and chemistry. So this is particularly an important area to address when you're looking at potential areas of variability. When we look at instrumentation and the type of instrumentation we want to use in order to develop a method that meets our analytical target profile or our goals, we need to identify instrumentation that will be fit for purpose as well. And of course, there are differences between different systems and differences between systems from different vendors. So again, we need to do a risk assessment around the different functionality or the different specifications of our systems. Now, the Mumbai College has recently acquired an analytical system, a Waters Acuity LCMS system, very similar to the configuration you can see in this picture here. This is an Acuity UPLC H-Class Plus system with a QDA mass detector. So the little unit on the bottom on the right-hand side is a mass detector. It's a very small modular mass spectrometer which fits with our Acuity systems. And we use this most often for analytical method development. So this is a quaternary liquid chromatography system for method development, has a number of options that allows us to develop robust methods using quality by design principles. And it's compatible with different software tools. As we mentioned, software tools can be extremely useful for conducting method development using a design of experiments and looking at a high number of different parameters and conditions. So at Waters, we typically will use either Fusion AQBD software or the Dry Lab modeling software from the Molnar Institute. So these are two softwares which are not Waters products, but we collaborate with other vendors to help us to provide solutions for our customers that are fit for purpose. And so just to highlight a couple of benefits of having a QDA mass detector on this system that, like the one that the Mumbai College have acquired recently. So if you have a hyphenated system like the LC QDA system, it provides specific advantages when you're doing method development. Some of them are highlighted on the screen here. For example, you can improve the detection limit versus UV. It provides more selective detection when you're looking at complex samples. Mass detection can help to confirm peak identity, helps in peak tracking. Of course, you can use a mass detector to look at those compounds which are not visible with a UV detector. And other aspects including detection of coeluting compounds and ensuring peak purity, for example. And just to highlight a few of those, because when you're developing a good method, you want to ensure, for example, that you have pure peaks, that you don't have coeluting peaks. And a mass detector can be extremely useful when in combination with some of these softwares that we're using for method development. So the Fusion software has, in fact, new functionality which will allow us to incorporate QDA data into the automated AQBD method development. So that's extremely useful to help us to identify, as you can see here, that the peaks are pure, that we don't have co-eluting peaks. And in this example as well, when you have typically in your UV chromatogram, you might have a peak which could be pure. It could have a slight shoulder on it. This is in peak 4. If you look at peak 4 in the top chromatogram, you can see that there's a bit of a shoulder on there. But with UV, you don't have so much information about what's behind that peak. If you introduce mass spectrometry, of course, you can see that in the bottom 2 chromatograms that we've identified that actually there are 2 compounds, completely different compounds, co-eluting at the same retention time. So if we add a mass detector to our method development, then this is a very quick way of helping us to ensure that we can detect and identify all of the peaks present in our chromatograms and ensure that we're separating those. And generally, when we're changing conditions during method development and our peaks are moving around, and particularly if we have very complex chromatograms, it's extremely useful to have mass detection present and be able to look at the masses behind some of those peaks to quickly identify which peaks are which. So again, just helping us to speed up the method development process and to ensure that we really have all of the information at our fingertips that we need when we're developing in order to develop robust methods. And finally here, this is a screenshot of the new Fusion Peak Tracker software. So just to illustrate the fact that if you are using UV as a detector, you're not necessarily going to detect all of the compounds present in your chromatogram. And you can see in the top chromatogram there, there's one peak at a retention time of 4 minutes that's not detected. But if you look at the lower chromatogram, this is the total ion chromatogram from the mass detector, there is actually a peak which is present at 4 minutes, which is picked up using the mass detector. And a nice feature of this new software from S-Matrix actually is that all of the information from both the mass detector, from the TIC and from the UV is collated in a global peak tracking method to allow the software to use all of the information from both the PDA and the QDA detector when carrying out the statistical analysis and evaluation of the different conditions evaluated and looking at the design space at the end. So it's a really nice new feature of the software which helps us when we develop methods using a quality by design approach. So just to give you an example now, this is a method which is an Ebastine API method published in the European Pharmacopoeia quite some years ago now. And as you can see, it's not a great method. It's a long method, 160 minutes runtime. And there are some poorly resolved peaks here. There's a very short retention factor here between the first peak eluting. The peak shapes are not great. And as with typically with older methods, with older approaches to developing analytical methods, it's very unlikely that the goals were set up front, that the method development and design was related to the actual production process and related to the critical parameters that they needed to monitor. So we can redevelop this method. And in fact, this method was redeveloped by one of our collaborators called Chromicent. And Chromicent are a contract analytical organization based in Germany, in Europe. And they redeveloped this method using the H-class, that system you saw earlier, and the QDA, but they used DryLab software. So DryLab software is a quality by design based modeling software produced by the Molnar Institute and works very well with our H-class system. And you can see in the bottom chromatogram, this is the redeveloped method. And so they applied all of these modern approaches to redeveloping the method. They set goals initially as to what they wanted to achieve with the method. They used the applied quality by design approaches to assessing the variability or the potential variability around the method. They considered sample preparation, they considered the instrumentation, and they used dry lab software then to help them to apply a design of experiments approach to developing the method. And if you're interested in all of the details of how they did this, there's a reference at the bottom of the page here, and we'll provide the slides afterwards so that you can have a look at this particular paper that was published and how they really went about this whole process in detail. But essentially what you can see in this bottom chromatogram is a greatly improved method. All of the peaks are much better separated. So the resolution between all of these peaks is improved. And the peak shape is also greatly improved. And of course, what this allows is a greater robustness to the method. When this method is used elsewhere, when it's transferred, along with all of the information that goes with this method, there's a much greater chance that there will be a successful transfer of the method, and that this method will be successfully used, implemented and used in a routine use. Of course, it doesn't stop there. And once the method is implemented in routine use, there needs to be trending of method over time to ensure that it continues to remain fit for purpose and give good quality results. Just one other aspect to add here, of course, you might have noticed that the runtime is much shorter. So the initial 160-minute runtime has been reduced to around just under 3 minutes long. And again, in terms of cost savings for companies, this is another really business driver for introducing these modern approaches to developing robust methods that will run with a much, much shorter runtime. So we did mention earlier control strategies and the benefits of implementing control strategies when we're developing analytical methods. And control strategies are used to prevent analytical variability. So that if you identify a potential area of variability that you cannot get rid of, that you can't reduce significantly enough, then you might need to put control strategies in place around your method. It will ensure the consistent quality output of the analytical method, and the fact that you really need to keep referring back to your analytical target profile or the goals of your method. You need to ensure that you're still meeting the original goals of the method. If you don't, then the method is essentially not fit for its original purpose anymore. So this is just an example here about some instructions that were written as part of an SOP relating to sample preparation. And in the first example on the left-hand side, you have the unclear method instructions, which just say stir until well dispersed. So this is fairly ambiguous. And then the example on the right-hand side, which is more controlled, stipulates stir no less than 30 minutes at a minimum of 2000 RPM using a 1 centimeter stir bar. So you can see that there potentially could be variability caused if all that you have available to you is stir until well dispersed. And different operators will potentially read this differently or interpret this instruction differently, and then potentially you'll have a different outcome, a different result. So it's very important, if there is an aspect of variability potentially, to ensure that method instructions are very specific and allowing you to control the way that, for example, the sample preparation is carried out. Another example here of a control strategy is relating to detection of peaks. So if you were developing a method, and this example is a peptide map with quite a number of peaks, as you can see in the top chromatogram, detected by the UV detector. The peak of interest, which is indicated by the arrow here, is coeluting with the peaks around it. And in this situation, it might be the case that the quantitation is not consistent. You might have interference, you might not be quantifying in a consistent way each time. And if you apply a different approach, and in the second chromatogram, in the lower chromatogram, they've applied another detector, which is the QDA detector, actually, in this case, to detect using single ion recording. It's a detection technique using mass spectrometry, the mass spectrometry uses, in order to just detect ions related to this specific compound. And by doing this, it makes it much easier to accurately and reproducibly integrate and quantify the peak compared to a UV approach. So again, a control strategy, by using another detection technique, which allows you to make sure that you are accurately and consistently quantifying a particular peak. And then finally, a final control strategy is an example, but also related to integration. If you have a chromatogram, such as the one you can see here on the right-hand side of the screen, there are potentially different ways to integrate the chromatogram. And so, again, this can cause differences in the data that's generated. And so it's very important in situations like this to not only to define the correct way to integrate peaks, but also to train users on how to integrate peaks. And in some cases, companies provide example chromatograms in their SOPs and pictures exactly how peaks should be integrated in order to be consistent time after time. So it's extremely important not only to design and develop good methods, robust methods that are fit for purpose, but also to ensure that you understand variability around the methods and that you put control strategies in place to avoid discrepancies and challenges moving forward. So coming back to this initial graphic from the USP 1220 proposed chapter on method lifecycle. It's extremely important to develop your initial goals, your analytical target profile related to your product, and then to ensure that the method is designed and developed using quality by design principles, that validation is conducted in line with your goals of the method, and that once that method is transferred into routine use, that the method is continually monitored over time to ensure that it continues to be fit for purpose. And if any changes are made to the production process, then a review will have to be conducted to ensure that the goals of the method are still valid and any changes required are put in place. So just to give you a little bit of information on where you can get some additional resources around method lifecycle management and the new guidelines which are proposed and in review. This is a microsite, a website which is available to anyone. If you search in your internet browser for methods.waters.com, then you will be able to see this site. There's a huge amount of information on the different stages of the proposed method lifecycle. And you can see, if you at the bottom of the screen, you can register yourself for updates. And anytime anything new is posted, then you'll receive information and updates. And one of the things available on this microsite that I think is particularly useful resource, if you're interested in this topic, are the podcasts. So we've developed a series of podcasts, and we're now up to series episode number 10, relating to a whole range of topics on analytical procedure lifecycle. And these podcasts are essentially interviews with leaders in the industry, so leaders in the pharmaceutical industry who have implemented or who have a very deep knowledge of the new guidelines which are proposed. And they contain a huge number of very, very useful insights into this whole topic. So you can either download those from the microsite, or you can also search for them on iTunes as well. So that concludes my presentation for this morning.
On behalf of everyone here, I would like to thank you for your most interesting presentation.
Thank you.
Dear delegates, the very aim of inviting a resource person who is working with a regulatory authority on global platform is to understand the forthcoming guidelines. If we can anticipate and prepare for future, we can survive in future. Now, without taking much time, I would like to throw open the session for discussion. Now, dear Stephanie, since we are running short of time, I would pick up few questions from the because attendees are being very inquisitive. One of the attendee, Ashish Sharma, has a question for you.
Okay.
Yeah, he is asking about what open source softwares are available for developing analytical methods and which software you are using in particular.
Sure. Good question. So in order, just as a background comment, in order to develop good quality, fit for purpose, robust methods, it's actually not essential or not necessary to use software packages or statistical software packages. It is possible to do it without. However, saying that, it's very useful to have a software package. It does help significantly. The software companies that we work with very actively are S-Matrix, and S-Matrix develops and provides a software called Fusion AQBD. A very nice software package which is compatible with Empower CDS. So there's a two-way data transfer into Empower software. So and for many customers who use Empower CDS, this is a big advantage. The second software that we work a lot with is DryLab. DryLab is a software from the Molnar Institute. And again, a very nice modeling software used to apply AQBD principles to analytical methods. There are others, and very much depending on the strategy you want to take. For example, Modder is a design of experiment software which can be used. So there are numerous ones, but we focus on Fusion and DryLab.
Okay. I think that settles the query of Mr. Ashish Sharma. Now, once again, on behalf of everyone, I would again like to thank Dr. Stephanie for the thought-provoking session.
You're welcome. Very happy to be involved. Thank you.
I'm pleased to introduce next speaker, Dr. Guillaume Bechade, who will speak on biopharmaceutical characterization through a regular LCMS analysis. Dr. Guillaume is working at Waters, and he oversees biopharmaceutical field marketing activities in Europe. Recently appointed to this role, he has been there for past 5 years at a large molecule market specialist in Germany, Benelux and Switzerland, helping industrial and academic partners to meet their challenge. He focuses on offering inspiring analytical solutions for the discovery, development, and quality control of antibiotics, oligonucleotides, and therapeutic peptides. Guillaume joined the Waters for last 10 years as an application chemist. He has an initial background in chemistry and obtained a PhD in LCMS-based proteomics, from the University of Strasbourg, France. Now, I would like to hand over to you, Dr. Guillaume.
Thank you. Good afternoon, everyone. Can you hear me okay? Can you hear me okay?
Yes, sir. Yes, sir.
Good. Thank you. So thank you for joining this session today, and many thanks to the organizers for welcoming me to this very nice e-conference. So my name is Guillaume Bechade, and at Waters I oversee biopharma activities in Europe from our headquarters in France. So my colleague Stephanie Harden has just shared with you the latest insights about small molecule pharmaceutical analysis. So let's turn now to the large biomolecule world. So in the next half hour or so, I'll be delighted to tell you why we believe that the biopharmaceutical analytical community can benefit from most accessible and deployable liquid chromatography and high-resolution mass spectrometry solution, and this for routine characterization and monitoring of biopharmaceuticals. So the biopharmaceutical industry is currently living profound dynamics. Biosimilars are spurring intense competition, and they are driving the need for much greater productivity, especially among developers and manufacturers. This in turn is fueling investment in new development and manufacturing technologies like greater MS downstream or continuous manufacturing. It also pushes towards modern analytical approaches like Stephanie has just described, quality by design or multiple attribute monitoring. And this can provide a very competitive edge to these people. Outsourcing the development and the manufacturing of biologics also continues to expand. Outsource partners work on a cost plus service model. So they are not earning revenue for the sales of the drugs being manufactured, but on a markup on the services they provide. This really creates competition, and they need to be as productive and as cost effective as they can be. Finally, the complexity of biologic is increasing, and as more and more products come onto the market, there is a strong need for better analytical tools that can provide and that can confirm the biophysical characteristic and identify the highly complex molecules with greater accuracy and greater precision. And not least, efforts are really being made to harmonize regulatory standards globally, especially with the International Conference on Harmonization, the ICH. Import-export rules are also in many cases favoring local production. So manufacturing and quality release testing really needs to be replicated in multiple geographies. And this is no exception to India, probably. So in terms of technology, supporting these trends is really important. And mass spectrometry in the biologics license applications filing has been increasing in the past 2 decades. You see here on this graph that from 2000 to 2015, almost every biological license application had mass spectrometry data in filing. In addition to that, the number of attributes measured by MS over the time has been steadily increased. The FDA from the US recognizes that additional MS methodology is seen within the quality control and comparability sections, which becomes more and more predominant. It appears that there is an evolution of the LC-MS usage from non-regulated environment to the highly regulated and compliant environment. So with biopharma, mass spectrometry technology is generally accessible through expert central or support labs in the companies. And these labs own the full characterization as well as the routine monitoring work. Many samples and lots of information needs to flow between these labs that are very busy and the other teams taking care of, for example, bioprocess development, stability testing, or QC testing. These situations create major bottlenecks, especially as business pushes to shorten time to biological license applications filing for the reason I have just explained. You see also this data integrity and GMP compliance needs that difficultly apply to all the core labs. The capacity constraints in central labs is very important, and the workload is strongly increasing, eventually opening the door to outsourcing. And all of this makes the support labs in the end wait for results, sometimes for a very long time. And the data needs to move across labs and eventually across geographies when all these departments are not in a central location. So that also creates bottlenecks. So for these reasons, when you ask how mass spectrometry can better be deployed beyond the core labs, people generally answer that they need a new type of instrument. and they don't need the highest specifications in terms of sensitivity or resolution or overall performance. They need really something that redefines the performance in terms of robustness of the instrumentation, the reproducibility of the measurement, the ease of operating the instrument and deploying the analytical platforms. They also eventually need a small footprint for the MS equipment. And last but not least, they really require informatics platforms that will help them to get actionable information that can help the scientists to take decisions. So based on this feedback from our customers, biopharma lab scientists, we have designed and introduced early last year the Bioaccord system that is a compact, robust benchtop, high-resolution MS system based on a time-of-flight technology that we incorporated with what we called smart MS technologies for maximum ease of use and accessibility. I will come back on these concepts very shortly. This platform is optimized for routine biopharma analysis at the protein, at the peptide, and at the release glycan levels. It's the first instrument that we have designed that is so workflow-driven with automatic data acquisition, automatic processing, and reporting that really facilitates the speed to results. And finally, this platform is compliant ready. It means that you can use it in a non-regulated or regulated environment to meet all the requirements. So what does it mean to be a smart MS-enabled system? So a little bit like the QDA is, that Stephanie has introduced before my presentation. We want to make things easy and convenient for the user. So it's a one-button startup system. In a single press, the system is powered on, pumped down, and will initiate an automated setup. This automated setup is consistent for all parameters and calibration procedures. And the system can be controlled and monitored through an intuitive user interface. A simple, clear indication of current status and any action required is displayed right on the panel, front panel of the mass spectrometer, which gives a very direct feedback to the user. And finally, we put in the background intelligent diagnostics that will help the system to check automatically the health and monitor for any problems and provide guidance for resolving them. If we have a closer look at the interface for the user in terms of computer, you see here the setup page for the instrument. So on the contrary of very advanced characterization MS platforms that require many clicks and many pages to be reviewed and parameters to be tuned, the Bioaccord system setup is very simple. You see it's a 3-button option, start, stop, or reset, that really streamlines the setup of the instrumentation. There are 4 options for this instrument to be used with: positive or negative ionization mode, low and high mass mode. So low mass would be used for peptides, oligonucleotides, for example, high mass more for proteins. Positive modes is generally for peptides and proteins, and negative ionization mode for oligonucleotides. On top of this, the system, as I said, has an intelligent health system with self-diagnostics. It means that if anything goes wrong in the system, the front panel will display a warning, like on the screen here. The system tells you to check that something is wrong with probably something that looks like a leakage. The computer part of the platform will tell you exactly how to solve this issue. And a little bit like a photocopier machine, it will tell you step by step how to resolve the issue. Step 1, open the door. Step 2, connect the leak sensor that has been detected as missing. And finally, close the door. Very simple steps. When the system detects that this has been done, it will recheck its health and give you a green light if the issue has been solved. If not, the level 2 in the problem-solving algorithm will be displayed and will help you further to solve the issue. In terms of software, we have based this platform on our unified platform that is really a software that oversees the workflow for the analysis by LCMs. It allows you to handle sample information, to create method and sample sequences, to acquire the data directly in the platform, to process the data, and finally to report and manage the information. So a single platform for all these steps. And as I said, this is a compliant-ready platform. So you have, as part of the software that is based on an Oracle database, all the tools that are required by the regulatory bodies to help you meet, for example, the 21 CFR Part 11 compliance guidelines. So you can have this platform installed on a network architecture with multiple systems. You can manage components libraries. You can define multiple users and assign different roles and rights to these users. The system can, of course, be qualified itself, the hardware part. Standard operating procedures can be implemented in the workflows as part of the system. And finally, all the reporting and data management tools benefit from safety features and electronic signatures that allow you to respect all these compliance guidelines. So let's turn to the application part now. As I said, this is a very workflow-focused platform that will allow you to do intact mass, subunit analysis, peptide mapping and peptide multiple attribute methodology, and release glycan analysis. For intact mass, this would eventually consider to be used for denatured protein analysis to get a mass confirmation of the protein or to assess for the proteoform heterogeneity. But you can also do native protein analysis, for example, to look at acidic and basic impurities of proteins using ion exchange chromatography directly coupled to mass spectrometry. For subunit analysis, also here you can do denatured and native. This would give you more access to structure information and eventually primary structure information and eventually post-transactional modifications, as well as some impurities if you use the native ion exchange. Peptide mapping and peptide monitoring will go much in detail on the primary structure information and PTM verification. And this is a very site-specific method for profiling and monitoring of these modifications. The release glycan analysis workflow will give you a direct access to the full glycan structure in terms of profiling and quantitation. And finally, you can also consider the intact mass workflow, not only for proteins or monoclonal antibodies, but also for synthetic biomolecules like oligonucleotides or synthetic peptides can be. So here you can do mass confirmation and impurity profiling eventually. So in the remaining part of the presentation, I'd like to give you some more practical examples around these applications, and starting with intact mass analysis. So here we cover the intact and subunit workflow. And this is really designed as a total workflow, as I said. So we can provide specific application-specific consumables and reagents in terms of kits that will help you to develop your methods and to assess the performance of the platform. We can provide, of course, the separation technologies that you may know, like the Acuity UPLC protein columns. Will it be C4 chemistry or BioResolve reverse phase polyphenol chemistry? This is for the reverse phase. And for native, we have a set of columns, like the size exclusion acuity UPLC protein column, or the most recently introduced bioresolve strong cation exchange column for charge variant analysis. The instrumentation would be made of an Acuity I-Class Plus system with a UV detector and the Acuity RDA, that is the TOF mass spectrometer that is part of this platform. And finally, the unified informatics. So let's have a look at the results. This here is what you get when analyzing an intact monoclonal antibody. We have analyzed here the monoclonal mass check standard that we typically use to assess performance of the instrument. And you see on the left-hand side a multiply charged spectrum of this monoclonal antibody. And when zooming on this charge species, you see that each peak is actually made of multiple peaks that are corresponding to the glycosylations. This multiply charged envelope can be deconvolved into a full intact mass spectrum automatically, and this is what you see on the right-hand side. This gives you the actual information in terms of the different glycoforms that your monoclonal antibody is made of. This information can be translated into a table where the mass will be displayed. You can confirm that the expected mass for your protein and your glycosylations is correct. You see that the mass error in the table is very low, so you get a very good specificity for the identification. And the relative intensities, relative composition for these glycoforms will be automatically calculated based on this spectrum. So for example, the first glycoform is representing 33% of your monoclonal antibody glycoforms. So this is one measurement, but in a typical industrial environment, you would need for very robust results and eventually batch analysis. So we made sure that with this system, you get a very, very good reproducibility with repeated injections. You see here the same sample that has been analyzed during 5.5 days that represents more than 1,000 injections on this system. And the consistency of the measurements here between the different glycoforms is really amazing. Across these more than 1,000 injections, we get on the relative composition, typically less than 2% RSD variation on the measurement. So really impressive results for mass spectrometry in a routine environment. Now you could say, okay, that's one sample on one instrument. What about if I have multiple instruments in my organization? Well, we made sure as well that you can get these very consistent and reproducible results across different systems. You see here the same sample that has been analyzed on 6 different systems. We repeated also the injection 2 times on system 1 and 6. And you get the same quality in terms of reproducibility. So less than 5% RSD on all these injections. That's really amazing when it comes to LC-MS analysis. Now that you have your analysis, of course, you'd like to communicate results in your organization. And the platform allows you to design reports and report templates that are really nicely customizable. So all the graphs, chromatograms, and pictures that I've showed you so far are actually extracted from the software and can be used in a report template. So you see on this example here that I have combined chromatographic data, would it be UV or mass spec chromatographic data? You have the raw spectra and the deconvolved spectra, tabled information, extracted ion chromatograms, and trend plots can be displayed as well. So a very nice workflow from sample analysis to results in a very automatic fashion and robust way. So that was intact mass analysis. Let's turn to maybe more specific information in terms of peptide mapping and peptide monitoring. So in this workflow, you will digest your protein into multiple peptides using specific enzymes like the trypsin. And using the chromatographic method, you will separate all these peptides on a reverse phase method. So we can offer here typical C18 chemistries like BEH, CSH, or HSS, depending on the type of peptides you want to your molecule is made of. All these peptides will be automatically analyzed using the same platform with a UV detector and the mass spec. And the same informatic platform can be used to process and report this information. Let's have a look to the results. So you see on the left-hand side, the separation profile for all the peptides in your sample. So it's much more complex chromatogram because you have much more species, but the software will integrate all these peaks, extract the MS and the fragmentation information, as you can do the analysis at both intact peptide and fragmented peptide, which will end up to the information that you see on the right-hand side with the coverage sequence automatically calculated. We have 96% of the whole sequence that have been confirmed here. And you also get this fragmentation information confirming the sequence at the amino acid level. So this is displayed thanks to the blue and red lines on the sequence, where fragments from the Y and B fragmentation profiles have been confirmed. And this is done using quite strict criteria with a mass tolerance of 10 ppm window. We only considered peptides that were more than 4 amino acids in length, so that explains why you have some gaps in the sequence here. And we have also a strict identification criteria, as we have only considered peptides that could be confirmed with free fragment ions. If you want to dig deeper in this data, the software allows you to display all the peptides that have been identified automatically. So you get the peptide sequence, eventually the modifications that an amino acid can have, like oxidation, deamidation, carbamidomethylation. And these modifications are automatically calculated in terms of relative intensities, relative composition. For example, the peptide 60 and 61 in the table here is the same peptide, but part of it has a loss of lysine. And this represents, as you see, 83% of this peptide. So you get immediately this information available in this table. The fragmentation viewer on the right-hand side confirms the identity of the peptide. So you see, we were able here to really read the fragmentation information and to build the sequence information on this fragmentation spectrum. So that's an additional confirmation and specificity parameter to help you to have a strong confidence in your data. So what I show you here for a loss of lysine can be also considered for any modifications that you want to look at. For example, an oxidized peptide. You see here an extracted information about peptide 21 that is present in the native form and in the oxidized form. So we have displayed here extracted ion chromatograms that shows the clear peaks for these two peptides. The fragmentation profile that confirms the oxidation and the localization of the oxidation on the amino acid sequence. And finally, we have repeated this injection multiple times, as you see in the lower part of this window. And we get here very repeatable results with an RSD variation less than 2% for this quite low oxidation level, 2.5% compared to the native peptide. So the quality of information is very strong here, and you can use all these criteria to confirm and ensure that your data has the best quality. All of this can be turned into a very routine workflow for typically quality control laboratories. And these people, they typically want to have a green or a red light when the oxidation, for example, is above a specified limit. You can program the software to do that. We have here monitored the oxidation of the peptide I have just mentioned, and we set limit checks that will warn the user when the oxidation level is higher than 10%, displaying the samples in yellow, for example, here. And it will give an error to the operator when the oxidation level is higher than 50%. So with this, you can really implement routine limit checks or routine critical quality attributes monitoring in a routine environment. Let's have a look now to the release glycan workflow. So this is a little bit more complex workflow because it requires a little bit of sample preparation that we can provide a kit for. We also provide the appropriate columns for this analysis that is based on hydrophilic interaction chromatography. And we here use the same platform, but with a fluorescence detector so that the glycans can be detected using the fluorescence signal on top of the MS information. And once more, we use the same informatics platform to perform the analysis. So typically, the sample preparation is a little bit tedious. Conventional methods make use of 2AB or 2AA labeling in a 3-step workflow. You need first to release the glycans from the protein using a typical PNGase enzyme. Then you need to label the glycans with these 2AA or 2AB tags. And finally, the glycans need to be purified. So you remove the protein and you remove all the reagents to keep only the glycans ready for the analysis. Typically, this requires 5 hours to 2 days of sample preparation. We have completely designed a new kit with specific methods that helps to streamline and to fasten the analysis in 30 minutes only. So the release glycan can be done in 10 minutes, thanks to a reformulated PNGase enzyme from New England Biolabs. We have designed a new label that is called RapiFluor-MS that can label all the glycans in 5 minutes. And finally, we use the GlycoWorks Helix microelution plates to purify the glycans in less than 10 minutes. So all in all, you can get your sample ready for injection in 10 minutes. The software has an automated workflow that will then integrate the chromatographic separation data and label or annotate all the peaks with the appropriate glycophore. So you see the end result here of the fluorescence chromatogram labeled automatically with all the peaks that could be identified. The MS signal is here to confirm the identity of the glycans that have been identified and can be used even for the lowest abundant glycan species. Like you see here, a 1.1% glycan can still present a very nice MS spectrum. So with this workflow, actually, we aim at really offering the protein scientist with the best release glycan analysis possible. So you get this sample preparation in less than 1 hour. We have very good sensitivity performance compared to instant AB, 2AB, or procainamines that are different labels that can be found on the market. We typically have at least a 2 times higher sensitivity in fluorescence and a 3 times higher sensitivity in MS, which really enables the workflow to be very good. We get reproducible chromatography. We get databases for automatic identification, the mass confirmation, as I said before, and a quick data analysis using the unified platform. But you see, maybe something is missing here to make it really perfect, is to make the sample prep part automatic. And we have worked on that very recently in partnership with 3 of the main automated platform providers like Andrew Alliance, Hamilton, and TCAN, for which we provide automated scripts that have been verified and designed by Waters people. We offer purposeful kits for all these platforms with the appropriate volumes. And all of this offers scalable sample processing from 8 to 96 samples at a time on these different platforms. And Waters was so excited with the Andrew Alliance solution that actually we acquired the company very recently to make it part of our total portfolio offering. Andrew Alliance has introduced a new pipetting robot very recently that is called Andrew Plus, that is really amazing. So you see the picture here. It's a very easy to use robot for which you rapidly can create protocols, and this does not require programming knowledge or a specialist. It's very versatile because you can scale and adapt the workflows by simply swapping the so-called dominoes in front of the robot, and the robot can recognize the type of domino and the type of consumable that you have positioned on the table. It's very easy to deploy because it's all based on a cloud software that will ensure maximum productivity and really minimize the downtime. And we provide the application knowledge for all of this, as I said, as we have developed and validated the scripts for that platform. So all in all, it ensures a much better reproducibility and traceability compared to the manual preparation, and it will maximize your lab efficiency and the resources. Of course, using a robotic platform needs to give the same results as a manual work, and we made sure that this was true. So you see here 4 different glycan forms that we have measured using the workflow that I have described to you. And we compared the manual sample preparation, the Andrew Plus robot sample preparation, and TCAN robot sample prep for 48 and 96 samples. And you see the results here are all spot on and very in good correlation between each other. So all in all, a very nice and automated workflow for N-glycan and release glycan analysis. So you remember I showed you this slide in the introduction, and we try to help the pharma companies to embrace the future and to in-source a multi-stage MS analytics platform and make these capabilities very close to the decision points. So in the bioprocess, in the analytical development, in stability labs or in QC labs. We really believe that with this, the lab throughput will increase dramatically, helping the segmentation of the full characterization work that still needs to be done by expert people in central labs, from the routine monitoring workflows that can be insourced by the bioprocess analytical or analytical development labs. It will dramatically reduce the wait time for these labs to get the results, because they can do their own analysis. It will reduce the risk because you will be able to run more, if not all, the desired samples on one platform in your lab. And finally, it will help develop and transfer methods between the different labs because of the very nice integrated informatics platforms we have. And this will really empower the non-MS expert users to run this technology very successfully. So here we are with the Bioaccord platform. As I said, it's been introduced early last year, and we're really already very successful with it. The platform has received very nicely 2 awards from customer boards. The Frost & Sullivan Awards, awarding the new product innovation for 2019, and the Select Science Award for the best new drug discovery and development product for 2019. So we are very proud of these 2 awards because they also come from our users. You can find more information on our website, waters.com/bioaccord. You can, of course, also contact us and Waters India for more information or for a demonstration on this platform, as this is really something good to experience. And finally, you can also find more collaterals and more information in application notes that are available on our website or on Select Science, as you see here, an application notebook focusing on mass detection, uncovering both the Acuity QDa and the Bioaccord system for biopharmaceutical analysis. With this, I thank you for your attention, and I'm open to question or using the chat or live. Thank you.
Thank you, Dr. Guillaume. I would like to express our appreciation for your interesting talk. Since we are running short on time, I would like to ask all the delegates and participants to leave their question on the Telegram chat group we have created for this conference. We'll surely get back to you. Dr. Guillaume, large molecule studies, proteomics, and biopharmaceutical characterization is a hot topic, and it has gained immense importance, especially in the current situation. I thank Dr. Guillaume for enlightening us on how to explore more using the current tools in hand. I would also extend my thanks to Prakash Babu for his continuous support, and also Prakash Mohanty from Water Corporation for eagerly replying to any queries regarding the arrangement of this particular session. I once again thank Dr. Guillaume and Waters India for their continuous support. With this, I would like to for next session, I would like to hand over to Dr. Bhavita. Dr. Bhavita, you may please continue.
Hello? Hello, Bhavita madam. Hello? Hello? Bhavita madam.
Please unmute her. Amol sir, I'll request you to please unmute Bhavita madam.
Can you hear me now?
Yes, sir, loud and clear.
Yes, yes. Yes, sir, we can hear you.
I'm ready to go anytime.
I would like Dr. Bhavita to formally introduce you, sir. Thank you.
Thank you.
Recording button.
Amol sir, can you see my screen? I'm sharing my screen.
I see the introductory screen, yes. I will be happy to simply introduce myself and give my talk.
Madam, please introduce to Dr. Arthur Frank. Hello, Bhavita madam. Hello. Hello, sir. Just
Hello,
wait for.
yeah, sir. I think we are facing some technical problem. Just for a moment.
That's fine.
Yes, welcome to the Global Challenges in Environmental and Occupational Health, a lecture that will be delivered by our great speaker, Dr. Frank Arthur. Dr. Frank Arthur is a professor in medicine at the Drexel University in Philadelphia, USA. Very well known to us because he has been coming to India during several occasions, a dozen of times nearly, where he would share his work on occupational and environmental issues that were being faced many a times. It's not only that, Dr. Frank is a figure who has been traveling all throughout the world, and he is in fact exchanging and delivering many lectures. In fact, frankly speaking, I should say, Frank emphasized to make his introduction absolutely simple. And this clearly shows his humbleness. So I wish to invite Dr. Frank Arthur to deliver his lecture on global challenges in environmental and occupational health. Dr. Arthur Frank.
Thank you very much, Professor Siobhan. It's a great honor to be with you at this international conference. Obviously, the world is a bit different these days because of the situation throughout the globe, but I understand we have many participants, and I'm honored to be able to give this lecture. Next slide, please. We are going to be speaking with you today about environmental and occupational health. And while health and medicine are very much linked, and medicine as a field of human endeavor has been around for thousands of years.
I would request Professor Frank to deliver his lecture.
Can you hear me?
Yes, sir.
Yeah. Did you hear my introduction, or should I redo it? No, I'll just continue.
Requesting Frank Arthur to deliver his lecture. I think sir is there.
I think Dr. Siobhan needs to mute her participation. So medicine is an old field. It goes back thousands and thousands of years in India and Asia.
Hello.
Hello.
Professor Frank Arthur, are you there?
Yes, I'm here.
Hello.
Madam, he has already started his lecture. Please unmute yourself now. Please.
Thank you. While medicine goes back many thousands of years, the first science that looked at workplace occupational medicine is relatively recent. An Italian physician, Bernardino Ramazzini, who was born in Italy in 1633, wrote the first textbook in occupational medicine in 1700. And he added one very simple question to the standard question that doctors asked, and that was, what is it you do for a living? And that opened up new aspects of understanding health and workplace environments. The general environment, what we might call environmental health, is a relatively recent addition to medicine. It is really only about 20 or 30 years old that we realized that hazardous materials did not stop at a factory gate. They would go out into the environment. People would bring materials home with them and could contaminate the household as well as the general environment. Next slide, please. So we need to recognize that the field of occupational medicine and environmental health has not been a part of the mainstream of clinical medicine. As you heard, I've had the good fortune of traveling throughout the world, and no matter where I go, the training and experience that physicians and health personnel get in occupational and environmental issues is quite limited. Next slide, please. There are some generic issues, no matter where you are in the world, that you need to keep in mind when thinking about these issues. The first is, of course, to assess workplaces and to assess the workers in that workplace with regard to their health. This is the essence of occupational medicine, is to link workplace exposures to disease and to assess how workplaces may do this. Then we have the issue of training and manpower, and how well do we meet the needs of the hundreds of millions of people at work. And then there's the question of doing research in this field. Next slide, please. Activities related to the workplace and workplace assessment are universal. As I said, they are done all over the world. However, it is mostly in America where we study environmental issues. And it was in America that the name environmental health was really added, and organizations are now called occupational and environmental health, and journals have added environmental to occupational health titles. Next slide, please. How much of an issue is this? If we look at the data that's available from around the world, we see that these are the kinds of numbers which are absolutely staggering. On a yearly basis, there are 100 million yearly injuries at workplaces. There are at least 100,000 deaths from injuries at workplaces. Not only do we deal with injuries, but we also deal with the issue of disease. And we have over 11 million diseases reported every year, with 700,000 disease deaths related to exposures from work. Next slide, please. We need to remember that farming is still the most frequent job in the world. More people are engaged in farming-related activities than any other kind of occupation. Nevertheless, we now know that more than half of the world's populations live in cities, which carries its own environmental hazards of crowding and appropriate housing. And in places like India and much of Southeast Asia, much of South America, farming is still the major source of exposure to people as they go through farming activities. Next slide. Farming is not just related to agriculture, but as we've seen in a number of settings where this is common, it includes ranching, which is the raising of animals, forestry, and traditionally fisheries come under the broad field of agriculture and agricultural health. Again, in some parts of the world, this is more important than others, and little work is done in agricultural health with a few exceptions. Here in the United States, for example, we have about a dozen centers funded by the federal government looking at agricultural safety and health, but otherwise, with a few countries like Sweden and elsewhere looking at this, it is not a common area of science. Next slide, please. There are a number of traditional settings for those of us like myself who do occupational medicine. Most individuals are employed either in company settings where they serve as company doctors for workers at specific workplaces, or in clinics.
Of science.
A number of government positions include people with occupational health backgrounds. Academia, at least here in the United States, is less than 10% of all such physicians. And then there are NGOs and sometimes labor unions that also hire occupational physicians. Next slide, please. I've just lost my slides. Thank you. The next slide, please. There is, in the area of training and manpower, there is a much longer tradition of occupational medicine in Europe than in the United States. This includes having hospitals that are devoted only to workers. There are a number of such workplace hospitals in a number of countries in Europe. Textbooks after that first one by the Italian physician were written in places like France and Great Britain, starting in the 1800s with the development of the Industrial Revolution. We find that there is very little training and workplace-related activities, particularly in the developing world. That is beginning to change, but only slowly. Next slide, please. How little training are we talking about? Well, here in the United States, where we have a somewhat different medical training regimen in the European model, which is also used in India and most of the world, is one goes to medical school after leaving secondary school. Here in the United States, one does an undergraduate 4-year college degree and then goes on to a 4-year medical training program. In the United States, where this has been measured, in the 4 years of training that physicians get, there are approximately 4 hours in 4 years devoted to workplace exposures. Now, this is an abysmal thought, given that most adults work, everybody is exposed in the general environment, and yet there is so little training. When I've taught in India and elsewhere in parts of Asia, even in Europe, I find there is very little training in occupational and environmental exposures that might lead to people getting sick. And over the years, I've been a physician for almost 50 years now. There are abysmal records in most medical charts of any physician asking about exposures. Next slide, please. Considering how many people work and how much of a relationship there is between exposures and disease, we note that the greatest shortfall of needed practitioners, again, as has been measured here in the United States, is in the field of occupational medicine. Elsewhere in the world, the numbers are also quite small. Next slide, please. For example, I looked up the data for India. Most of the training in India in occupational medicine, and I've taught in postgraduate level courses in all of India with your 1.3 million or so people and hundreds of millions of people working, you have approximately 1,000 doctors, only 1,000 that do occupational medicine. In terms of those individuals who do measurements at workplaces, a critical piece of information about exposure, in all of India, there are probably somewhere in the neighborhood of about 100 industrial hygienists. This is a field that is starting to get some recognition, and as an interesting field for students to go into, this might be something that should be thought of. But there is very little ability to go measure workplaces. And even when measurements are taken, there are few laboratories available on the Indian subcontinent to do the analysis of the measurements done at the workplaces. I've on occasion had to send specimens out of the country to be analyzed elsewhere. Next slide, please. Research in occupational medicine is often an afterthought. Most clinicians working in this field are essentially concerned with taking care of the workplace and the workers in that workplace. And so little research is done. There is generally very modest government funding anywhere in the world, perhaps with the exception of the Scandinavian countries and Finland, where they are much better about funding this. Here in the United States, the funding levels for research for occupational health issues is somewhere less than 10 cents per worker per year. That would be a fraction of a rupee. So there's very little funding to do research. And progress comes very slowly in terms of workplace-related research because of this. And yet we deal with a lot of serious problems, as you'll see in a little bit. Next slide, please. So as I said, there's very modest funding here in the United States. There's some research done in Europe, some in South Korea at a few institutions, a little bit in China. But again, very modest when one considers the vastness of workplace exposures and the problems that exist compared to other issues that get looked at. Next slide, please. So what are some of the important issues in occupational and environmental health these days? I'm not going to spend a lot of time on this, but clearly to my mind, the most serious problem facing everyone in the world is the first item listed here, which is climate change. What we see with the pending changes in climate is that we are going to change the ability to access water. I know in India and cities like Chennai, they already have severe water shortages. Johannesburg in South Africa has a similar situation, so that climate change will bring about shortages of water. It'll change growing seasons. It will change where pests are found. In cold climates where pests tend to die out over the winter, the warming of the earth will see that the pests don't die out. And so vegetation will be adversely affected. I think that We already see the effects of pollution that leads to climate change. We see data of 2 to 3 years lost in the average lifespan from significant pollution, as we see from data, for example, coming out of China. It's also going to change employment and lead to underemployment with these changes. Plus, as we see more and more mechanization around the world, fewer people are going to be employed in traditional tasks, and perhaps only those with advanced training to fix equipment are going to have access to good employment. Around the world, we still have the problem of migrant labor. We have people from many countries migrating to other countries to undertake employment. That puts them at great risk for abuses of many kinds. That's a whole area of concern for those of us that think about occupational health issues. And then an issue that is relevant to China and many, well, not so much China, but to India and a number of other countries, is the whole issue of child labor. In India, the government will admit to about 14 million children that really should be in school in the workforce, but as many of us recognize, the number is considerably higher. Next slide, please. What we also see is that industries move from one set of countries to others. A number of years ago, when I started in this field, in my early days as a physician and scientist, we had movement around the United States. We had businesses moving from states that had stiff environmental regulations or had high salary levels, and people moved to other companies moved their factories to other parts of the United States. What we've seen in the last 30 and 40 years is not only do they move within countries, but they move between countries. The push factors are regulations, both in the work and in environmental settings that companies don't want to meet, and labor costs in some countries obviously are higher than elsewhere. The pull factor is to the countries that have less regulation, that have cheaper labor with a lot of unemployment, and so they can pay low wages. In those countries, there's sometimes a shortage of hard currency, so they welcome outside multinational companies to come and bring money into their country. And then national issues of industrialization. Again, India is a country where the government is very much looking to increase production of Indian goods. China is doing something similar. They want the Chinese to buy Chinese goods, as the Indians want to have Indian goods bought. And here in the United States, we hear about buying American goods, but a lot of them are no longer made here, particularly in some areas like textiles, clothing, and such. Chemicals are not so often made here either. Next slide. So I'm going to go through what I thought about and thought were some major areas of concern. They're listed here. Occupational lung diseases, which are a major problem in the world, pesticides, radiation, a relationship of workplace health to general primary care, ergonomics, which is a field that looks at man-machine interactions, person-machine interactions, injuries at workplaces, which we've pointed out are a big problem, hearing loss, eye injuries, and psychological factors at work. Next slide, please. Occupational lung disease, which is an area that has occupied me professionally for most of my career, is an old issue. Ramazzini wrote about occupational asthma, for example, from agricultural activities. We know that there are mining-related diseases that go back and were written about 500 years ago, but we constantly keep seeing even new exposures From Turkey, we've learned that people who want to have blue jeans, denims that are modified in terms of their coloring, they use rocks to wash the color out, and people have been getting ill. Jewelry polishing is another area where grinding goes on. And a major new one is what we call artificial stone countertops in parts of Europe, Australia, here in the United States. Countertops made out of silica-containing material have made workers quite ill with silicosis. The other issue, and this is one that I published on with some Indian colleagues and others fairly recently, is we understand that talcum powders around the world, something that's widely used in India, throughout Southeast Asia and elsewhere, can lead to asbestos disease because these talcum powders can be contaminated with asbestos. Next slide, please. Pesticides are another material that are widely used around the world and are a source of great illness among those who work with them. Pesticides, we now know some of them can cause cancer. We know some of them cause sterility. We know that companies have tried very hard to sell large quantities of pesticides and don't always educate farmers as to the ideal use of such pesticides. For example, a number of years ago, I was doing some work in Egypt and I learned that they were using collectively some 300,000 pounds of pesticide to grow crops in Egypt, but the crop yields were not particularly good. One of the problems they found out was that they were using too much pesticide and it actually prohibited plant growth. When they cut back to some 10%, some 30,000 pounds of use, they found that the crops did much better, but companies were not about to tell the farmers that, having wanted to sell larger quantities. Unfortunately, we've learned that in some countries, India being one of them, that pesticides are often used to commit suicide. Farmers in places like India, but here in the United States as well, face very significant issues, economic issues in trying to make a living growing crops or raising animals. Another area of my work over my career has been in the field of agricultural safety and health, and have studied those issues as well. And despondency among farmers is not an uncommon problem around the world. Next slide, please. Radiation has become a more important issue. We certainly have the experiences of the last few decades of Chernobyl and Fukushima putting out large quantities of radioactive material into the general environment, affecting obviously not only the workers who worked at these facilities or had to go in and clean up these facilities, but creating enormous hazards for following environmental exposure. The other issue that concerns me that I wanted to raise is waste disposal. As we talk about using more nuclear energy around the world, something that some countries are embracing, but others are shutting down nuclear facilities. Nowhere in the world has anyone figured out how to do waste disposal of the spent fuel rods that are used, and as they build up over time, are a source of potential significant problem in the future of what we do with all this radioactive waste that is out there that may end up causing problems both at the site and if they are released into the general environment. Next slide, please. In some settings, healthcare is provided to workers. I've been at a number of occupational facilities in India and elsewhere, where primary care is given at the workplace to workers, sometimes to their families as well. We don't do so much of that here in the United States. There's a bit of that, but then primary care is something that is needed everywhere in the world. Obviously, some places are in greater need than others. So even those working in occupational health settings not only should understand something about the workplace, but also the general environment and general primary care, and integrating into workplace settings primary care for workers and their families is something that is still being thought about. Next slide. I mentioned ergonomics before. It's what we call man-machine interactions. And the question there is, what are we seeing around the world with regard to issues of musculoskeletal problems? And we see those particularly with the growth of the use of computer screens and keyboard use. We see it in manufacturing, repetitive motion in building things like automobiles. Workers will get musculoskeletal problems due to repetitive activities, something that we never saw when people used something older that some younger people may not appreciate, called typewriters. But with modern computer work, and I'm sure even many students have noted when they do a great deal of typing, that they may end up with some musculoskeletal problems in shoulders, wrists, and fingers. This is really a fairly modern phenomena and is again a worldwide or a global issue that we worry about in occupational health. Next slide, please. Hearing loss is a very common result of work. We see that again around the world. Many workplaces are quite noisy, but again, there are some special issues of noise. We see it with people riding motorcycles. We see it with in those parts of the world where gun enthusiasts shoot guns. In India, we've seen problems, obviously, at the time of Diwali with the use of firecrackers and people having all kinds of injuries, but among them is the issue of hearing loss. Next slide, please. Occupational health psychology is really quite a small field. It is one with very few training sites here in the United States, very underappreciated with very few personnel around the world. But yet, on a trip to India last year, I was asked to visit at the largest steel mill that operates in India. And they asked me to give a talk to the safety and health people, and the specific area that they wanted me to talk about was psychological aspects of workplace health. I found that really quite interesting. It's not an area that I myself have done much research in. I know some of the people here in the United States who do work in that area. I have a colleague at Drexel who looks at some of these issues. But to be asked out of any number of topics to talk about psychological issues at the workplace meant that it is getting some increasing importance. It also is important, I think, in other settings. We see at some of the factories in China with the making of electronic materials, for example, the suicides that have been reported from overwork. And here in the United States and a few other countries, Australia, some work in the UK, looking at what we call first responders, people like health personnel on ambulances, firefighters and police, and some of the psychological issues that are related to those kinds of activities. So that's again, another area that one needs to think about in this broad understanding of workplace and environmental health issues. Next slide, please. As I mentioned in my opening remarks, we now need even to consider pandemics and workplace and environmental issues that have raised. We see literally the whole world has been shut down and modified by the appearance of a virus that wasn't known about a year ago, and yet has affected millions of people around the world, has shut down countries, has created disruption all over the world. We recognize that until we have either better treatments or that we have some kind of appropriate vaccine, that everyone in the world will continue to be at risk, and how this affects our daily interactions in our environment or our ability to go to work and be safe in those settings is going to be greatly modified. We also learned that there is an unfairness to who gets ill, because we find that poorer people have to do the kinds of jobs that don't allow them, for example, to work from home, such as I've been able to do for some months now. They work in very tight workplaces. We know that food processing facilities here in the United States have been a particular problem, but we have a whole set of new considerations because of what has happened with this new virus. Next slide, please. So what are some of the international organizations that give advice or look at issues of environmental and occupational health? First, we have the World Health Organization and its sister organization, the International Agency for Research on Cancer, IARC, which is based in France. These organizations, both the WHO offices in Geneva and IARC, give recommendations with regard to how to work safely in workplaces. We have the International Labor Organization. Again, they've been around for many, many decades and give advice about safe and healthy workplaces. We have the International Conference of Occupational Health, a sort of NGO group that looks at these issues. We have true NGOs like the Collegium Ramazzini, named after Professor Ramazzini, that look at health issues and put out pronouncements about safety and health issues, banning materials at workplaces. We also find that there are some countries that assist others. As I mentioned, the Finns and the Scandinavians do a lot of research, and they are willing to travel around the world and assist others in carrying out improvements in workplaces. I've seen colleagues from these countries literally in many places around the world. We do little now from the United States. I hate to say this, but 30 years ago, the United States was probably the leading country in the world in terms of workplace safety and health issues. We no longer can claim that, and we have regressed greatly while also not helping others around the world. Next slide, please. There are any number of international journals and places where one can read about occupational safety and health and environmental issues. And I've got this phrase, good and bad. I've been a scientist for a long time. I've published in the literature. I've probably done reviews of, for many, many dozens of scientific journals over the years. And I hate to say this, but there are some very legitimate journals that I would be proud and have been proud to publish in. And then there are some journals that are absolutely fronts for poor research or for research that is done simply for the purpose of confusing issues about workplace safety and health, and at the end of the day are there to protect bad companies who do bad things from being sued or being accused of truly bad habits. And so when individuals go out and read journals, I think one needs to educate oneself about which journals are sort of true and honest and which ones are run by groups of individuals that are apologists for companies and for their bad behavior. It may take a while to sort this out, but again, journals will have these kinds of reputations. Next slide, please. So this all leads me to bringing up this issue of ethical challenges. Go back one, please. Back one slide. Please go back. Well, all right. The slide's not up, but I'm going to talk a little bit about ethical challenges with regard to workplace safety and health. There are governments that are supportive of industry, as they should be on occasion, but they also can get in the way of protecting their own workers and workers around the world. For example, there's something called the Rotterdam Convention. It's about 125 or 30 nations around the world that meet regularly to put on notice hazardous materials that countries and their workers should be aware of, and yet individual countries can block this kind of recognition. It requires a unanimous vote of all countries, and India, for example, has blocked banning asbestos, chrysotile asbestos. There are many countries that put out pesticides that they know are harmful, or put out pharmaceuticals that don't meet proper standards. And then we have something that I call science for sale. This goes back to the journals that I was talking about. There are scientists who literally for a price, and the price can be quite considerable. In one area that I've done work in, looking at the hazards of asbestos in friction products like brakes on automobiles and motorcycles, one company has paid out some $40 million to scientists to come up with favorable research. And then at the very worst, we have something called corporate spying, that people have gone out to try to infiltrate NGOs and look at scientists who are doing work that might cause them a problem. Looking at the slide that's up now, we have various legal issues. How well do companies follow rules in workplaces? Again, we know that in some countries, workplace inspections are rarely done. Here in the United States, even though we have federal standards, the number of investigators available to go out to workplaces means that if every workplace would be inspected, it would literally at this point take 100 years for the small number of inspectors to look at every workplace. We have companies that fight compensation for injured workers. Again, they will hire people to fight against what really seems to be appropriate compensation. And then there's the lack of applicability to some groups that they fight to have regulations applied to them. Not only do we have regulations in the workplace, but we have regulations in the general environment, which are much harder to enforce, and governments often do not wish to do so, especially if they want to have industrial production increase. We see that in Russia, China. India now has become a country where environmental regulations are not used to protect the people in the general environment very much. The next slide, please. Another issue that is growing around the world that we need to keep in mind is small-scale operations such as mining, illegal mining that goes on in unsafe conditions. We have electronics extraction when all of the equipment that we use, the computers and cell phones, when they get recycled, go to Africa and Asia to have extraction of the precious materials, putting people at work. We have illegal logging going on. We have poaching of animals. And then we have other groups that come to mind, like those who do scavenging in waste dumps to recycle materials there. Next slide, please. So just in closing, I want to say that the provision of high quality occupational medicine services and environmental health understandings needs to be improved all over the world. We really have a need for more such activity, more such training of physicians, more young scientists to do research. For someone like myself who's at the end of his career, I've tried to make my contribution, but so much more is needed. And I hope that that will come to pass. So thank you very much. I think we have some time for questions. You're on mute, Dr. Siobhan.
Sir, yes.
Oh, that's better.
One. Yeah. So an enlightenment talk, and we have some few questions that have come up in the chat box. Mayuri Udupa has asked if there are industrial workers, we are exposed to chemicals, more or less over here we get cases of asthma. So would occupational medicine help them out? To what extent? Occupational medicine in cases of asthma. How can we manage them?
Occupational asthma is a major problem in workplaces. Chemicals, many other things. I have trained occupational physicians throughout my career. Asthma is a major problem in the world. Much of it is not related to workplaces, but there are many, many occupational asthmagens, and any physician working in occupational medicine, some of their training clearly comes in understanding occupational asthma. It runs everywhere from certain wood products to many chemicals to agricultural products that will cause asthmatic responses. It's a major issue that needs to be addressed.
Yes, thank you. That was a question from Mayura Udupa. There is another question from Leesh Hirani, who says that can we have preventive health programs in companies and industries to help the people?
Yes, I was mentioning before that many companies are now looking at preventive or looking at primary care. Well, part of primary care is to do preventive health. There are some companies that I'm aware of here in the United States and Europe that provide preventive services that go beyond the actual workplace exposures. They would provide good nutrition in the company dining rooms. They would provide exercise facilities. They would do some kinds of testing at workplaces. During flu season, they will provide vaccinations for the flu. So clearly, workplaces and medical programs at workplaces are a potential venue in which to do preventive services.
Yes, sir. Thank you, sir. And I think probably this is something that has been overlooked. In terms of technology, strides are taken. But when it comes to occupation, beyond that occupational health, we talk of occupational hazards, but occupational health hazards, that would be more challenging. And now in this present situation, I think it would be further magnified if proper precautions are not taken. Sir, you have enlightened us and have thrown light on many aspects. You've not even spared the journals over there. You've mentioned about journals, you've mentioned about the NGO, you've mentioned about all the waste and how much we should take proper precautions. Overall, I think you have covered up and an eye-opener lecture on global challenges in environmental and occupational health. Sir, on behalf of the wish to thank you for this very nice session. In fact, in the chat box, I've got many of the comments saying that it is excellent, it is very informative, much neglected topic, occupational health medicine. When it comes to medicine as an aspect, occupational hazards we know, but what about the medicines related to it? So I think the topic was well covered, and we thank you all, sir, for your good delivery of this lecture. And we hope to find ourselves in better conditions, and we hope that there would be some improvement through government policies. You have spoken about the different nations, the different national policies, and let us hope for the best. At this note, I thank you, sir, for joining us from Philadelphia. Thank you, sir.
Thank you very much for the opportunity. And one last word.
Yes, sir.
When I was a young scientist, I worried about the idea that over my future career, all the real problems would be solved. As I'm now at the end of my career, I realize that not only have we not solved most of the old problems, but there are far more problems that we've never recognized, and that any young scientist or any student thinking about the future, there will be plenty of work in this area of environmental and occupational health and exposures that will make for a wonderful career. So thank you very much for the opportunity of joining you, and thank you for your comments.
Very true, sir. Thank you very much. And I assure you that young researchers and participants who have joined this program would definitely take this point, and a much neglected one, but of course, in days to come, may be aggravated. As you have said, you have done so much of your research, and now you foresee that there's lot to be done. We will take this as an inspirational and a motivational talk. And with your blessing, sir, thank you very much.
Thank you. Goodbye to all.
Thank you. We now move to the next. I wish to thank Dr. Aparna for the play of the slides. Thank you, Aparna Saraf. And with this, I would say that the first day of the conference, the international conference, has come to an end, the first day. And I'm sure the participants are very, very eager for the second day, because the second day begins with oral and poster presentations. very much keen on them, and you'll be given an opportunity to present. They will be judged and on parallel chat boxes. So you be alert and put your best, all the best for your performances, all those who are participating, young researchers. Now, going with the schedule, tomorrow 9 o'clock, we expect you to join. And as we proceed through the oral and poster presentation, it will be followed. We have 3 lectures for the day. One will be delivered by Professor Gulab Khedkar. Another one will be by Dr. Jacinta D'Souza. And then we have Dr. Anja Jangnikil, who would be talking on atomic absorption spectrophotometer. So we have, and then lined up would be the valedictory. So hoping to see you all. At the same time, there is a small reminder that we have a feedback form of this particular conference, day 1. And it has been displayed. I think the link has been put on the Telegram group and on the YouTube. So I request all the participants to please fill up the feedback form and forward it. Of course, it will count for your e-certificate as well. On this note, I thank you all. And also at the same time, I am absolutely sorry for holding you up at times due to the technical snag. Of course, this is an online conference and certain things would be definitely improved. Saying so, I wish you all a very good day, and that is all. Thank you very much. We meet tomorrow morning. Thank you.